my name is Sofie Salama, and I head up what we affectionately call the wet lab part of David Hausler's research group. And I recently gave a talk to the first year students in their BME or bioinformatics boot camp about a major part of our research, which is looking at neural development, our Brain Engineers project, and looking at the development of human-specific features of brain development. And so, so that they wouldn't have to hear the same thing over again, I thought I would talk about another major project in the lab, which is really looking, it also focused on evolutionary biology, but really interested in the role of retrotransposons and the machinery that we use to regulate them and how that influences the evolution of new gene regulatory elements. And another motivation was that this project has a lot of collaboration with Benedict's lab. So since we were both speaking today, it seemed like a nice thing to do. So how do I get this? Okay, here we go. So as you know, we have sequenced the human genome for many years now. And, you know, in our lab, one of the things that we're really interested in is comparative genomics and using that to understand what makes us human. And one of the striking findings of the initial sequencing of genomes was the finding that actually quite a lot of our genome is actually composed of transposable elements. So these are elements that are sort of considered parasites of our genome and replicate themselves and move around. And in the human genome and primate genomes in general, the majority of these transposable elements are in fact retrotransposons. So retrotransposons are elements that necessarily go through an RNA intermediate in their life cycle of inserting themselves in the genome. So they get transcribed, they get in fully functional retrotransposons, they get translated, and then use a reverse transcriptase and integrase to copy themselves into the genome, and thus they take advantage of the host, us, our gene, protein, and expression machinery. And so these retrotransposons actually pose a threat to ourselves because they tend to actually insert themselves into actively transcribed parts of the genome. And thus, if they insert themselves either within a gene, in a protein coding region or a regulatory region, that can be bad and in fact could be potentially lethal. However, they also can become part of our genome and become an essential part of our function. And so this recent review by Cedric Fauchette shows many examples that have been documented by which either a sequence derived from retrotransposons either becomes essential part of gene regulation or post-translational regulation. And I saw that Manny's in the audience, and he could probably go on a lot about NMD exons that are retrotransposon derived. But in our lab, we've been really focusing a lot on how actually the cellular machinery that silences retrotransposons to thus prevent them from jumping around in our genome, how that regulatory mechanism develops, and then how that can lead to changes in gene regulatory networks. So how do we prevent retrotransposons from running amok in our genome? Well, one of the major ways is through a large family of transcription factors called the KRAB zinc finger proteins. And this is a large family of modular transcriptional repressors where they take advantage of arrays of zinc finger domains, which can denote DNA binding specificity, and then also have a KRAB domain, which is a protein interaction domain that actually recruits this factor CAP1, which itself is a nucleating factor for a number of proteins, which together lead to generating repressive chromatin, both through histone and eventually through DNA methylation. So this is a large family of genes. There's about 400 in the human genome, and in fact, 190 of these have emerged recently within the primate lineage. And back in 2010, there was a couple of studies that established that a specific KRAB zinc finger protein was both necessary and sufficient to repress a mouse endogenous retrovirus and its mobilization in the mouse genome. And this led to the hypothesis that the dramatic expansion of primate KRAB zinc fingers was driven by a role in controlling retrotransposons in primates, in addition to mouse. And we were very taken by this and set out Frank Jacobs and Dave Greenberg when they were postdocs and graduate student respectively in the lab, did a really nice body of work where they demonstrated that repression of two different classes of retro elements, the SVA elements and the primate line 1 elements, were respectively repressed by two different zinc finger proteins, both of which happen to have emerged in the primate lineage. And so since that time, we've wanted to sort of broaden our look at this and look more systematically. And to do that, Max Hausler, and then later in collaboration with Jason Fernandez, really developed the UCSC Repeat Browser, which it really enables systematic analysis of genomic data on retrotransposons. And the way that this works is basically we generate a consensus for each subclass of retro elements. So in this example, this is a particular primate specific line 1 element, the L1PA4 elements. And then basically every single instance of that element on the genome gets mapped back to this consensus. And then you can map any genomic data back to these consensus sequences. And so the nice thing about that is in essence, you are sort of aggregating data. So in this case, here we're looking at ChIP-seq data from two landmark studies that performed ChIP-seq on over 100 in each study, KRAB zinc finger proteins. And then what we're seeing is what we so-called peaks of peaks. So for every individual instance where a particular KRAB zinc finger gave a ChIP-seq signal, we're mapping it back to the consensus element. And then those when you add all of those up together, you get these nice peaks showing that multiple different KRAB zinc fingers are binding to these line elements. And then we can take one data type here. These are individual zinc finger ChIP-seq experiments. And now in this third row, we're looking at where CAP1 chip maps on this L1PA4 consensus. And we can see that while there's a bunch of KRAB zinc fingers, that proteins that bind, only those in the 5' UTR actually correlate with this CAP1 co-repressor binding. And then we can also, so then we went on to determine that these peaks most correlate with ZNF93, which I already mentioned, and then another ZNF protein, 649. And then we can also look at other data. In this case, we're looking, so this CAP1 data is in human embryonic stem cells, which is an early embryonic cell type, which is actually a time in which these retro elements tend to be active, and it's sort of a dangerous time for their mobilization in the genome. And we can see that, in fact, 649 and ZNF93 are two highly expressed KRAB Zinc finger proteins in this important cell type. And so, so basically, we feel that these two proteins are the major KRAB Zinc fingers that are important for CAP1 recruitment to repress L1PA elements. And so, you know, then the next question, we already looked at this for ZNF93. So how does ZNF649 recognize DNA, and how has that evolved? And so one of the ways that we look at this is by using the motif that we've identified in using the repeat browser analysis to generate a reporter in which we put that segment upstream of a luciferase reporter, and we look at the ability of introducing ZNF649. In this case, we don't do this in human cells. We do it actually in mouse embryonic stem cells, which contain CAP1 and the other repressive machinery, but don't contain any primate-specific zinc fingers. So we can test the ability of 649 to affect the expression of luciferase, and we can either do ZNF649 or specific orthologs of ZNF649. And in this case, Natalie Philippi, undergraduate who's now a research specialist, found that while the human and orangutan versions of 649 can repress luciferase activity, the luciferase reporter, Gibbon 649 does not. And so that suggests to us that ZNF649 acquired the ability to repress L1PA elements at this point in evolution. And so we can also look to see how the line elements respond to the ability of 649 to repress them. And by both looking at sort of how comparing different L1PA elements. So I should backtrack for a moment. So line elements are characterized in these characteristic waves where, you know, a version of the element dominates and can move through the genome, and then that gets repressed, and then a new version emerges, and so on and so forth. And the naming convention is such that the older elements have higher numbers, which get progressively lower until you get to the version of line 1 that's currently active in the human genome, which is called L1HS. And so if you look, you know, starting with the L1PA4 element that I showed you is effectively repressed by ZNF649, you can see that as you go from L1PA3 to L1PA2 to L1HS, that you accumulate these mutations in the consensus binding site. And this binding pattern suggests escape from ZNF649 evolution. And so this is now summarizing a lot of work that we've done looking both on the protein side and on the L1 sequence side to really understand the interactions between the KRAB Zinc finger and the retro elements that it can bind in detail. And down here in this bottom panel E, we're showing a cartoon. So ZNF649, human ZNF649 has 10 zinc fingers, and these amino acid codes are indicating the amino acids that are implicated in DNA binding for each zinc finger. And then this is the consensus binding site that we've determined for ZNF 649. So based on this analysis, we kind of have a model for what's going on. So ZNF 649, interestingly, is actually an ancient zinc finger that is shared with between human and cat and pig. But looking at the line elements at about the emergence of the divergence of old and new world monkeys, the line elements underwent a major rearrangement that led to a new 5' UTR sequence. And shortly after that, or a bit after that, at the divergence of the lesser ape Gibbon from the rest of the great apes, ZNF 649 actually gained a third finger, and that seems to have been critical for it to be able to repress to repress LMPA4 elements, which then gradually escaped regulation of 649. And so this leads to the question of why did ZNF 649 ever exist in the first place? And given that current line 1 elements actually are not repressed by 649, why is it still conserved? And so one clue to that is comes from work that was done completely independently, where they were doing a screen for pluripotency factors, and actually ZNF 649 came up as a factor that was required for maintenance of pluripotency. And so in our lab, to explore this a little bit further, so, we've been doing our work in pluripotent stem cells, and I told you that ZNF 649 is highly expressed in pluripotent stem cells, and that's where it seems to be important in regulating these line 1 elements. So we took advantage of a so-called CRISPR-I human pluripotent stem cell line, in which this line harbors a doxycycline-inducible dead Cas9 KRAB. And we introduced guides to repress ZNF 649 expression. And so in the presence of doxycycline, you no longer have expression of 649. And we did RNA-seq, and we found that a few genes were down regulated. Gratifyingly, one of the most dramatic was ZNF 649 itself. So this is kind of the positive control, but then there was many genes that were upregulated, and this is those that had a significant p-value and at least 4-fold induction of expression. And so we see a number of genes that are upregulated, and interestingly, several of these have important roles in patterning and induction, for example, in the case of SOC17 and endoderm induction. And so what
you just showed us?
I'm sorry, yeah?
Could any of those other downregulated genes have been responsible for the upregulation you were seeing, or could you eliminate them as possible confounders?
We have not eliminated them as possible confounders, and certainly amongst the upregulated genes, many of them, for example, are themselves targets of SOC17, because the SOC17 is a master regulator transcription factor. So one of the things that we've been looking at we've been mostly focusing on the upregulated genes is, do they have L1PA elements in their promoter regions or nearby? And also, do they have ZNF 649 binding sites and ZNF 649? Is it able, do we see ChIP-seq signal? And so that's what I was just about to get into. So for example, so one of the things that we wanted to know was for the genes that are de-repressed upon ZNF649, do any of them have L1PA elements associated with them? And interestingly, what we find is a significant enrichment in a de-repression in genes that contain an L1PA2 element in their UTR. So, but in addition, we see, so that suggests that actually that by virtue of having a line 1 hop into the gene, we are actually pulling it into this ZNF649 gene regulatory loop. Interestingly, we see other genes, including SOX17, which do not have an L1PA element, but do have a strong ChIP-seq signal, and also have a sequence that kind of loosely matches the L1PA4 binding site. And so, so in this 649 ChIP-seq data, you see a strong signal of SOX17. And interestingly, ZNF649 is the most enriched gene in SOX17 ChIP-seq data, suggesting that there might be some regulatory relationship between these two genes. Another master regulator of development, the NODAL gene, also has a ZNF649 ChIP-seq. And when we made a NODAL reporter, we see that in fact ZNF649 is able to repress its expression. And interestingly, for NODAL, the human, and in this case, we've also made the human orang ancestral version of 649, are both able to repress the nodal promoter, but the orangutan 649 and the Gibbon 649 version of 649 are not. So, so basically, this, together, these data suggest that the ZNF649 line 1 arms race has rewired gene expression, both by virtue of so the evolution of line 1 elements and the gain of these three of this additional finger have swept up new promoters into this ZNF649 gene regulatory network, and then these L1, by virtue of L1PA2 jumping around the genome, we have new genes that seem to have been pulled into this pluripotency gene regulatory network. And it seems that this has now been selected to help them repress differentiation. So such that even though 649 no longer plays an important role in repressing line mobilization, it does seem to still have this important role in pluripotency. So just because for the new students, we're still working on further elucidating the role of 649 in line 1s, as well as primordial germ cell and embryonic stem cell gene expression regulation and the evolution of that gene regulatory network. More generally, we're interested in the role of recently active retrotransposons and the KRAB zinc fingers regulating them in models of both pre-implantation with the so-called naive stem cells and post-implantation primed embryonic stem cells, which is the data I've shown you today. We're also interested in performing a comprehensive analysis of KRAB zinc finger evolution and function in the primate lineage, taking advantage of some of the new genome assemblies and cactus alignments that have been generated. And this is in collaboration with Benedict's lab. We're exploring, and some of that work, especially using the new ape and old world monkey genomes, has revealed interesting evolution of both SVA and endogenous retroviruses. And so we're interested in exploring that. And then finally, recently, again with Benedict's lab, taking advantage of these really high quality assemblies and cactus alignments to look more broadly across vertebrate genomes to examine patterns of retrotransposon evolution and function. And so just to wrap up, this is the work of many people. I mentioned Max Hausler as the who really helped us make the Repeat Browser. Jason Fernandez was a postdoc and now project scientist who really sort of spearheads our efforts. Christoph, Natalie, Paola did many of the experiments that I showed you. Saul is our bioinformatician extraordinaire, does all kinds of great stuff. Marina has really been helpful with on some of these new primate assemblies, and then you know, other collaborators and funding. So that I think now I can make this bigger so I can see you guys again, and happy to take any questions.